So in order to talk about neurodegenerative diseases or diseases of the nervous system that are related to aging, it's important that we're all on the same page about some kind of basic neuronal anatomy, as well as how action potentials are generated, and different functions of cells in the nervous system, as well as different functions of brain regions. And so over the next couple lectures, I'm going to focus on making sure we're all kind of on the same page about that. I'm going to start by talking about some basic neuron anatomy and how action potentials transmit signals from one neuron to the next. And so a neuron is a polarized cell, which means it has two distinct sides. It has dendrites and it has an axon. And so dendrites are these branch-like structures here, and their main function is to receive signals. Those signals are then integrated in the cell body, or the soma, which houses the nucleus and other organelles, and then those signals can be transmitted down this long tubular structure called the axon, whose job is to send or transmit the signals to other neurons or other cells. And axons are normally wrapped in a protein called myelin, which helps insulate those axons and conduct the electrical signal that they're sending. And so it's important to note that not every single neuron looks the same. In fact, neurons are very highly diverse in both their morphology, or the way that they look, as well as their function. And we know that there's a direct kind of correlation between structure and function in all of biology. And so usually the different structures of neurons correlate with their functions. And so you can see here a cortical neuron, which has rather short dendrites in red and a really long axon in blue, extending out to here, and a Purkinje cell, which is the exact opposite. It has this huge network of dendrites to send signals, and then one single axon, which comes off of the image down at the bottom. But regardless of what function they have or what their ultimate structure is, all neurons do have these axons and dendrites. And all neurons also have this unique ability to change their membrane potential. And this is something that's relatively unique, as I said, to neurons and to muscle cells. And so all cells have a membrane potential. And what that really means is that there's a difference in the voltage or the electrical potential between the inside of the cell and the outside of the cell. There's a difference in voltage from inside to outside. And most cells can't really do anything about this, whereas neurons and muscle cells can. They can change their membrane potential. And these changes to membrane potential are what leads to the action potentials that we're familiar with in neurons. And so the resting or the unexcited membrane potential of a neuron is somewhere around here at -70 millivolts, which means this is pretty negative, and that negative number means the cells or the neurons are more negative on the inside than they are on the outside. And as you can see here from this graph showing the membrane potential of a neuronal cell, neurons can change this, the electrical state or this membrane potential through a process of depolarization, where they get less negative, and or hyperpolarization, where they get more negative. And so through depolarization and repolarization and hyperpolarization, neurons can change this membrane potential. And they do so using voltage-gated ion channels that exist in the plasma membrane of the neurons themselves. And so there are two kind of important voltage-gated ion channels in this process. There are potassium channels, which move potassium ions, and sodium channels, which move sodium ions. And what I'm going to bring your attention to as well is that both sodium and potassium ions are positively charged. So when they're moving, whichever direction they're moving is usually becoming more positive, right? And so in a normal resting neuron, the resting membrane potential, as we just said, is -70 millivolts. It's more negative on the inside, or the intracellular side, than it is on the extracellular side. You can see those little negative and positive signs here to help you keep track of what's going on. And so under these conditions, both of these channels are closed. There's no movement of potassium and there's no movement of sodium. There's just an established negative 70 millivolt membrane potential. When the neuron receives a signal to fire or a triggering event happens, there's kind of a slow depolarization or a slow change, making that membrane potential more positive. You can see that here in the graph. And this is due to kind of a slow opening of the sodium channels. Sodium can come in, and sodium is positive. So it changes the membrane potential to be more positive on the inside. Right, and so in that resting state, both of these channels are closed, and the resting membrane potential is negative 70 millivolts. Once a triggering event happens, and there's this slow depolarization which occurs, it remains slow as sodium ions come in until it reaches this dotted line here. And this dotted line is what's known as the threshold potential. And at this threshold potential of negative 55, the sodium channels all open. And rather than a slow depolarization, sodium floods into the neuron. And there's a fast depolarization you can see here as a peak, which is indicated by this peak. And this is what generates the action potential. So you've got a resting membrane potential of negative 70, slow depolarization up until negative 55, and then the sodium channels open, sodium rushes in and depolarizes that membrane, generating an action potential. And so after the action potential fires, and it reaches this kind of peak, most positive state, that depolarization continues until about positive 30 millivolts, right? At positive 30 millivolts, another change happens to kind of these voltage-gated channels. And the sodium channels close, and the potassium channels open. And now, all of those positive potassium ions rush out of the cell, making it mean the positive charge is moving out, the cells gets more negative and repolarizes as the potassium rushes out of the neuron. And this continues to happen all the way up until about negative 80 millivolts. And then those voltage-gated calcium channels are triggered to close. And at this point, the neuron is kind of ready to fire another action potential. And what you'll notice is that negative 80 millivolts, when the channels close, is a little bit lower than the resting membrane potential of the neuron at negative 70 millivolts. And this is done by design, so that action potentials can't be just fired one after the other. There's a little bit of a recovery period where the cell has to move back from negative 80 millivolts after repolarization to negative 70 before it can fire another action potential. And that keeps the neurons from kind of firing more action potentials than they really can. And so those action potentials are important, and the generation of that electrical signal and it traveling down the axon is essential for neurons to work. But unfortunately, electrical signals can't travel from cell to cell. They can't pass from one cell to the other unless those cells are connected. And in the case of the nervous system, that's not true. At the ends of the axons, there are axonal terminals or synapses. And synapses are considered a type of junction where one neuron communicates with another or where a neuron communicates with another cell. But what you'll notice is that at this synapse here, the axonal terminal does not actually touch the receptor sites or the receiving neuron. There's a synaptic cleft or a little bit of a space in between the axon of one neuron and the dendrite of another. And that means that electrical impulse can't pass directly from this axon terminal to this dendrite. So there's sort of a step in between. And at this synapse, there's sort of three main parts, right? There's the presynaptic neuron in purple, there's the synaptic cleft or the space, and then there's the postsynaptic cell. Most of the times this is another neuron, but it can also be like a muscle cell. And so in order for that electrical impulse to move across a synapse, it has to be converted into a chemical signal first. And so the way that this works is by triggering release of neurotransmitters. So action potentials travel down axons to axonal terminals, which you can see here. And that action potential triggers the opening of calcium channels at the axonal terminal. Those calcium channels and that influx of calcium into the cell triggers the release of what are called synaptic vesicles that are filled with small molecules called neurotransmitters. And those neurotransmitters get released into the synaptic cleft. They can bind to receptors on the postsynaptic cell, neuron, other cell. And when they do that, they actually trigger the opening of sodium channels. And when sodium rushes into the cell, as we just talked about, the cell becomes depolarized, and now this postsynaptic cell can fire another action potential. Right? So one action potential comes down, triggers influx of calcium, which triggers release of neurotransmitters. Neurotransmitters can bind to this next neuron, triggering the input of sodium by opening the sodium channels, which depolarizes this neuron and propagates or passes on that action potential. And so there's a conversion of electrical signal in the form of an action potential to chemical signal in the form of neurotransmitters to another electrical signal in the next neuron. And you've likely heard of neurotransmitters before. There are many different types. For the purposes of this course, I'm going to focus mostly on acetylcholine and dopamine because those are particularly involved in pathology of Alzheimer's and Parkinson's disease, which are both age-related and neurodegenerative. But you can see here that there are several different classes of neurotransmitters. There's about 50 to 100 different types, some of which, like glutamate and GABA, are made from amino acids. Some are neuropeptides or proteins, and some of which originate kind of as a cholesterol molecule, like dopamine and serotonin. And all of these neurotransmitters act on particular types of neurons and exert particular effects. But as I said, we're going to focus on these two, and so it's important to know their structures and functions because they're going to become important as we talk about both Alzheimer's and Parkinson's.